Hello everyone. Welcome to the rapid revision for the chapter Principles of Inheritance and Variation. And I see there are some people already waiting for us to start. Shri Trisha, Jivita, Shri Vidya, Akshaya. Hello everyone. For all of you who don't know me, I am Nupur Khera. I'm Dr. Nupur Khera and right now I am living in Switzerland and working here. So I am really hoping that you all have gone through the videos, or at least you have gone through the chapter, and then this rapid revision will be very helpful. Because here, we have tried to include the most difficult questions and also different types of questions, that you can expect in your examination. Okay, so let's begin. So the first question, and whenever you answer the question, just write down one and then whatever option is there. Then it's easier for me to see, how many of you are following. So among the following characters, which one was not considered by Mendel in his experiments on bee? You see, this is such a simple question and a direct question, and it was asked in the year 2017. Okay, so what is the right answer? So Mendel studied different characters, so which one he did not study? Is it A, trichomes, glandular or non-glandular? Is it seed, green or yellow? Is it pod, inflated or constricted? Or is it stem, tall or dwarf? So what is the right answer? Quickly write down in your chat box. Yes, so all of you wrote the right answer, which is option number A. Perfect. So then let's continue. The law of segregation of characters postulated by Mendel can be related to the presence of two genes for each character in a somatic cell, each of the two homologous chromosomes during meiosis, presence of both genes on the same chromosome, or none of the above. So, what is the right answer for question number 2? I start to get the answers. So, I'm waiting for everybody of you to answer. So, the law of segregation was the second law that was postulated by Mendel. Perfect. All of you gave the right answer. A gamete receiving only one of the two homologous chromosomes during meiosis. And one thing that you should note down here is, that this is what law of segregation can be related to. This was not the law of segregation. Mendel just said about factors. At that time, he did not. Okay, so this you should be clear. Sometimes they can twist the question in that way. But the option B or the option second is right. A gamete receiving only one of the two homologous chromosomes during meiosis. Mendelian factors, later known as genes, are present on chromosomes. Okay, which is the chromosomal theory of inheritance. So what is the right answer? There are three genes A, B and C. Percentage of crossing over between A and B is 20%, B and C is 28% and A and C is 8%. What is the sequence of genes on the chromosome? So what is the right answer here? And sometimes, if you get stuck or if there is any confusion, just keep on asking simultaneously, okay? I can always drop you some hints or we can work the question out together. So what is the right answer for question number 3? So I will wait till I get the answers, and now I start getting the answers. But I am waiting for more answers. Good. Perfect. So all of you are giving the right answer, which is option number A. So, in these questions, always start with the maximum percentage. For example, here they tell you that the distance between B and C is 28%. So, first it's B and C, and then in between it will be A. So, the right answer is option A. Always start with the maximum percentage. See, here there are just 3 genes. Sometimes they can give you even more. So, try to always start with the maximum percentage and then keep on going slowly down. Very good. Next question. Number of autosomes in a sperm is? The number of autosomes in a sperm is it 2, is it 44, is it 23, or it is none of these? So what is the right answer? So just read the question properly, okay? You have enough time in the exam. Just don't get negative marking by not reading the question properly. Okay, read the question properly. What I used to do, when I gave my exams, I used to always underline in the question, like, okay, they are asking about autosomes, they are asking in sperm. And then automatically, you can get the right answer because the questions are generally easy. Good. So what is the answer for question number 4? I start getting some answers, but I'm waiting for more. So everybody try to answer, okay? Even if you don't know the answer, just tell me I don't know. Okay, then it will be more interactive. Okay. Some more answers I want. In life you always want more. You want a higher rank. You want more marks. I want more answers. Come on, everybody try to answer, and if you don't know the answer, just write it down. I don't know. Good. Keep on giving me answers. See, we are here for you, okay? So just try to give me as many answers as you can. Okay, so for now, I have got only 2 answers from Karthi and Srividya. And both of them are saying B. Akshaya also says it's B, which is like the second option, I guess, 44. Okay, so what are they asking you? They are asking you number of autosomes. What are autosomes? Humans have 23 pairs of chromosomes, out of which 22 pairs are autosomes and 1 is sex chromosome. Okay, for example, a normal cell has ploidy 2n. There will be a process called as meiosis that will take place where? This meiosis will take place within the sperm or in the egg, and then 2n will become n. So what is 2n here? 2n is 46, n is 23. Out of this 46, 44 are autosomes. Now can you try to answer? Option B was wrong, guys. So can you now try to answer based on these hints? What will a sperm have? A sperm will have total of 23 chromosomes because it has half the genetic material. Okay, that is why 2n is always converted to n plus n during the process of meiosis. And this n and n will come together to restore 2n during fertilization. Can you now try to answer number of autosomes? Yes. Very good. Now I start getting the right answer. The right answer is option number 4, none of these. But now you have to write down the real number. What is the real number of autosomes? Okay, you wrote the option D, but what is the real number of autosomes? Quickly write down in the chat box. so that I know all of you have understood it. So please be clear about this. They can ask you about the sperm, they can ask you about the egg. Okay, this was one of those tricky questions, where you can easily get negative marking. That's why I said read the question properly and always underline the important things. Humans have 23 pairs or total 46 chromosomes, out of which 22 pairs or 44 are autosomes. One pair or two chromosomes are the 6 chromosomes. So they are asking you about the autosomes in the sperm. Good. Very good, Shri Trisha. All of you, just write it down. So all of you who wrote B, 44, you were half right, okay? You were writing about the normal cell. But they have asked you about the sperm. That is how they will, trick you in the question. So always read it properly. Good. Now I see a lot of you are telling me it is 22. Perfect. Good. Then the right answer is option 4 and let's proceed. An allele for hemophilia will pass from father to all his sons, 50% of his sons, all his daughters, or 50% of his daughters. So tell me what is the right answer for question number 5. An allele for hemophilia will pass from father to all his sons, to 50% of his sons, to all his daughters, or to 50% of his daughters. For this question, you should definitely know what hemophilia is, okay? Or at least, okay, I'll drop the hints later. Let's see how many of you can try to answer question number 5. Always give me the answer, and if you don't know, write down I don't know so that we can discuss, okay? You're here to learn. Answer for question number 5. An allele for hemophilia will pass from father to. Good question. It's a very good question. That is why, this genetic disorders from this chapter, Principles of Inheritance, that will contribute a lot of questions. And again, if you don't know the answer, just write down don't know so that I can proceed with the explanation and we can try to understand what is happening. Good, I start getting some answers. But I want more. I have received the answer from Shri Trisha. Very good. Good, I start getting the answers. Perfect. See, all of you are giving me the right answer. Why? Because hemophilia is an X-linked disorder. So this will be the father. Okay, and when the father, for example, marries, let's say, a normal mother, this gene will definitely go to all his daughters. Why? Because a daughter always has X chromosome and the daughter will always take the X chromosome of the father. Okay, and this type of inheritance is also called as criss-cross inheritance, right? I'm sure you all know this. Like from the father, the allele goes to the daughter. Okay, and when it goes from the mother to the son, this is criss-cross inheritance. Good. So all of you gave the right answer, which is option number 3. An allele for hemophilia will pass from father to all his daughters. Good. Then let's proceed. Following is a pedigree for albinism. Okay, small a is albinism. What is the probability of 2-1 to be a heterozygous? This individual here. Try to read the question properly. Take your time. Write down what you need to write down and then give me the answer. Okay, for pedigree analysis, never rush. That is the key to solving the pedigree analysis. This is a pedigree analysis for albinism. What is the probability of the 2-1 person to be a heterozygous? Tell me the answer for question number 6. You have been given that albinism is small a, okay? Pedigree analysis, you can definitely expect some questions from this chapter because pedigree analysis is the most asked from this chapter. So question number 6, what is the answer? And again, if you feel like you don't know, then just let me know. But you guys are doing amazing. Very good. I'm very happy with all of you. I start getting some answers, but I'll wait till I get answers from all of you. Good, this I like, 6B but not sure. Good. See, that is how you have to practice, okay? Even if you are not sure, it's okay. We are here to learn. But some more answers will be very nice. Okay. So, let's try to understand the question. Okay, following is a pedigree for albinism, which is an autosomal recessive disorder. Somebody else wants to answer, they can really answer. Who all is there? Akshaya is here. Srividya is here. Maybe I am getting the answers a little bit late, but it's okay. So, Karthi, Jivita, all of you. Okay. So here they say albinism is an autosomal recessive disorder. So you see, these are the two parents and they produce an offspring that has the disease. So definitely both of them should have a small a and small a, and the other one should be capital A, and that is why they have gotten also unaffected individuals. Now what you have to do? You have to do nothing but do a cross of both the parents to find the probability. Okay, what will be the population of children that will be observed? So this is what you will get when you cross them. Okay, so these are the 4 possibilities. And again, sometimes there is this confusion. Yeah, there are 4 possibilities, but they have just shown 3 children. So what do we assume? You have to assume nothing, guys. That is the probability of 1 in 4. If you do your maths, if they say the probability of getting is 1 out of 4. For example, if you talk about the diseased individual, the probability is 1 out of 4. What will be the probability out of 3? Then you have to calculate. So don't get confused by the fact that they are giving you only 3 kids here. Or maybe they can even give you 5 kids or 6 kids. But the probability you will always calculate like this. Okay, now what is the question? The question is, what is the probability of this individual 2-1 to be a heterozygous? First of all, you have to see what is this individual. This individual is phenotypically normal. How many of here are phenotypically normal? 3 of them are phenotypically normal. Okay, out of which 2 are heterozygous. So what is the probability? The probability is 2 out of 3. The right answer is option 3. Is it clear? Is it clear for all those who did not give me an answer yet and for all those who said B? You have to always take the population. Then you have to see what is the individual. For example, 2-1 here is not diseased. Okay, so the possibilities are 3. out of which 2 are heterozygous. So that is why the right answer is 2 out of 3. So you have to give me thumbs ups. If you have attended my lecture, you know I'm greedy for thumbs ups. Is it clear? See, the question is they are asking you what is the probability of that individual to be heterozygous. Okay, so always read the question very properly. Try to underline the important things. So I'm not getting any responses. Maybe I'm getting the responses late. But for all those who couldn't answer or for all those who said option B, is it clear? Good. Finally, I get a yes ma'am with smiley and thumbs up. But I want more, guys. Guys, be active. Okay, this is your time to be very active. Your exam is quite far but very close. Okay, and I trust me, if you have followed through all these questions, even if you are not able to solve all of them, but you have understood it, you know, you can really solve all the questions in the exam because we have tried to take all the different types of questions that are asked and they are actually towards the difficult side. Okay, good, perfect. So the right answer is option 3. Next question, very easy question. Pattern baldness is a sex-linked character, sex-controlled character, sex-limited character or Hollandric character. What is the right answer? Quickly tell me the right answer. See, this is one of the easy questions. Okay, so very straightforward, but then, you know, they will confuse you with this sex-linked, sex-controlled, sex-limited, Hollandric. Yes, the doubt. Okay, I'll just quickly go back to clear the doubt. Yes, capital A is homozygous. Okay, that is the thing, no? That is the basic genetics. Capital A, both the alleles are same. That is why it is homozygous. Small a, both the alleles are same. That is why it is homozygous. Okay, both capital A and small a are both homozygous. Only when the alleles are different, guys, one dominant, one recessive, then it is heterozygous. See, no, so that is the thing, no? What you also have to see is that this individual 2-1 is non-affected. Okay, that is the trick, that is the hidden trick in this question. Everybody will answer it 1 by 2, you know, but then it will give you negative marking. You also have to understand what is the probability of individual 2-1 to be heterozygous. The 2-1 individual is normal. If you say that the probability is half, and how are you calculating half? 2 by 4. In this 2 by 4, you also have one of this individual. And you cannot take one of this individual because the probability of small a of the individual 2-1 is 0. Because this individual is unaffected. It's a very good question. Who asked? Kutu Mahi. You asked this question. It's a very good question. I'm very happy with this question. Why is it not half? Okay, because if these two are heterozygous, the probability of getting is 2 by 4. No, but here they are already telling you in the question that this individual 2-1 is unaffected. Therefore, this individual cannot have this genotype small a. Okay, therefore you have to consider only these three because only capital A small a are phenotypically normal. 2 of them are carriers, but phenotypically they are completely normal. Is it clear? Kutumahi, and for everybody else, it's a very, very good question that you asked. It really shows that you understood the concept. But pointing you out towards the individual 2-1 and the individual 2-1 cannot have this small a genotype. Okay, because it is normal. So this though anyways you will not include, and that is why your probability is out of 3, and out of 3, 2 are possible. Is it clear or should I repeat it again? I can repeat it again. Yeah, Salma, there was buffering. So can you just now again click live? Because now it's streaming again, I guess. Okay. Okay, then it's fine. Okay guys, there was some technical issue. It was buffering or something like this. So coming back to this question here, I hope it was clear that why the answer is 2 by 3 and not 1 by 2. Okay, you cannot take this individual here. And I see a lot of thumbs ups that it is now clear. So let's proceed to the next question. Question number 7. Pattern baldness is a sex-linked character, sex-controlled character, sex-limited character, or holandric character. So what is the right answer here? So quickly tell me, this is one of the easiest questions that they can ask you, and they will ask you, because why? Because here it's very confusing, sex-linked, sex-controlled, sex-limited, holandric. Okay, but you have definitely studied this. So tell me what is the right answer for question number 7. And I see answers coming up. And everybody of you is answering option A, which is the wrong answer. Pattern baldness is not a sex-linked character. I hope you know the difference, between what is a sex-linked character, what is a sex-controlled, what is sex-limited. You can always go back to the genetics videos, that we did, but I will quickly explain you in brief. So first of all, you have to understand that pattern baldness is an autosomal. It is not an X-linked character, okay? The example for X-linked characters are for those genes that are present on the X chromosome. But pattern baldness itself is a sex-linked character. Okay, it is not a sex-linked character because it is present on the autosomes. Then is sex-controlled character. What is a sex-controlled character? Just listen to it very nicely, okay? What is a sex-controlled character? A sex-controlled character is one whose genes are present on autosome, but it is influenced by the sex or it is controlled by the sex. For example, pattern baldness, okay? The genes are capital B, capital B. Okay, capital B is bald in both males as well as in females. Okay, then small b is non-bald again in both males and females. But capital B, small b is bald in case of males and it is non-bald in case of females. And why is it that? This is because testosterone is present in males that will make this as bald. Is it clear? Is it clear? And then what is a sex-limited character? A sex-limited character is a character that is again autosomal, but it is only in one sex. For example, the Adam's apple. All the men have Adam's apple. It is only limited to the sex, but the genes are present again on the autosomes that are expressed only in men. Another example of the sex-limited character is breast and milk production in females. But you have to remember that sex-controlled and sex-limited are present on the autosomes. Good. See, now everybody is answering option number B. Very good. I hope it is clear. So, pattern baldness is a sex-controlled character. This difference between sex-linked, sex-controlled and sex-limited should be very clear, okay? Both sex-controlled and sex-limited are present on the autosomes. Good. So, the right answer is option number 2 or option number B. Next question. Question number 8. The number of different types of gametes produced from a plant with genotype capital A, capital B, small b, capital C, small c is? How many gametes, how many different types of gametes will be produced with a plant with this particular genotype? So what is the right answer? I hope you remember the formula, okay? Don't just start randomly making the gametes. If you don't remember the formula, I will tell you the formula, but try to answer yourself. Good. I start getting some answers already. I want some more answers. Just try to answer, okay? And this is one question, you know, that you can answer in seconds if you know the formula. And what is the formula? It is 2 raised to the power of number of heterozygosity. Okay, if you don't know this formula, please write it down. They will definitely ask you this question. Very good. All of you are giving me the right answer, which is option number 2, answer 8, because 1, 2, 3. 3 of them are heterozygous, so 2 raised to the power is 8. Now, sometimes what can happen is they can even ask you which gametes. Okay, how will you do that? Don't just randomly. There is this method called as fork line method. So how will you do? You will do capital A, small a. Then again you will divide this into 2. You will do capital B, small b, and here as well capital B, small b. And then again here capital C, small c, capital C, small c, capital C, small c, capital C, small c, and this will give you all the 8 gametes here. Okay, for example, these are the 8 gametes, and they can definitely ask you this question, so don't take this very lightly. You can very quickly form all the gametes with this method, which is also known as the fork line method. Okay, so if you don't know, I'm hoping that you already know this, but just in case if you don't know, this is the way, you know, wherever there is heterozygosity, just make a bulge and then you will get the right answer. Very good. So the right answer here is option number 2, 8 gametes. Question number 9, and this is an Ames question, very similar question but with a twist. How many different types of gametes can be formed by F1 progeny resulting from the following cross? Capital T, small t, cross capital R, small r. It's exactly the same question with a twist. So what is the right answer for question number 9? Use the same formula. This is an Ames question. What is the right answer for question number 9? You gave the right answer. All of you gave the right answer for the previous question. I start getting the answers now. How many different types of gametes can be formed by F1 progeny resulting from the following cross? Good. So everybody answered correct, which is option number A, 4 gametes, because again the heterozygosity is 2. So here, you know, they just confused you by saying a cross of capital T, small t, cross capital, and you will be like, what? We've never seen such a cross before. It's nothing, you know, but 2 plants where 2 different characters have been crossed together. So don't get very much confused. Just use the straightforward formula whenever there is this question about gametes. Good. Let's proceed. assertion and reasoning type of questions will be also asked. Okay, so that is why we have tried to include this question. So the assertion is cross of F1 individuals with recessive homozygous parent is a test cross. And then the reason is no recessive individual are obtained in the monohybrid test cross. Always try to do what? Try to see if the assertion is true or false. Then try to see if the reason is true or false, and then proceed to see, if reason is the correct explanation for the assertion, you will get the right answer. So question number 10. What is the right answer? So you need to know what is a test cross, and it's a very easy question. So if both assertion and reason are correct and reason is correct explanation of assertion, then it is option A. Both assertion and reason are correct, but reason is not the correct explanation of assertion. It is B. If assertion is correct, reason is incorrect, it is C. If both assertion and reason are incorrect, it is D. Don't get confused by the structure of the question. Okay, just try to see if assertion is true or false, if reason is true or false, and what is the right answer. And if you are not able to solve, then you know what you have to do. You have to write down in the chat box that I am not able to solve the question. Okay, I start getting the answers. Okay, so a lot of you are answering B, which is if both assertion and reason are correct, but reason is not the correct explanation of assertion. So what is assertion? Cross of F1 individuals with the recessive homozygous parent is a test cross. This is true. No recessive individuals are obtained in a monohybrid test cross. So if you know what is a test cross, you also know that test cross is used to see if an individual is this or if an individual is this. Okay. But if the individual is this and it is crossed with small t, what will you get? See, I got Jeevitha. You answered the right answer. See, when you cross these two individuals, what are you going to get? You will get capital T, small t, and you will get small t. Okay, so you will also get the recessive individual in a monohybrid test cross. Here they are saying no recessive individuals are obtained in the monohybrid test cross. So the reason is false. Is it clear? Yeah. Now a lot of you are giving me the option number C as the right answer, which is very good. But for all those who answered B, is it clear? You do get recessive individuals in a monohybrid test cross when you are crossing a heterozygous individual with a recessive one. You get half tall, half dwarf, which is the recessive one. Is it clear? So the right answer here is option number C. Assertion is correct, but the reason is incorrect. Is it clear? Give me thumbs ups. For all those who answered B option, some of them have retracted the answers. Okay, I'm hoping that it is clear, okay? Always try to think. It's just a monohybrid test cross they are asking. So you can always see, you can cross this and this and you can cross this and this. Okay, got confused with the options. That is why, if you're getting confused, what you have to do is always write it down next to the assertion if it is true or false. Okay, here it was true. Then write down next to the reason if it is true or false. Here it is false. You will never get confused. Okay, I know these assertion reasoning types of questions sometimes can be, what was that? What was this? If you write it down, it's straightforward. Okay, good. Then let's proceed to the next question, which is question number 11. In a test cross involving F1 dihybrid flies, more parental type offsprings were produced than the recombinant type offsprings. This indicates what? The two genes are located on two different chromosomes. Chromosomes fail to separate during meiosis. The two genes are linked and present on the same chromosome. Both of the characters are controlled by more than one gene. Read the question properly, read the options properly. If you know the concept, it is just a 1 second answer. Otherwise, just read it very nicely before you tell me the answer for question number 11. So what is the right answer? Question number 11. And if you don't know, just write down don't know. In a test cross involving F1 dihybrid flies, more parental type offsprings were produced than the recombinant type offsprings. This indicates what? I start getting the answers and I'm happy. Perfect. See, I'm so happy that all of you are answering the right option, which is C. Okay, the genes are linked and they are present on the same chromosome. That is why more parental type offsprings are produced than the recombinant types. This is linkage, which was discovered by T.H. Morgan. Next question. Don't get scared by the vastness of the question, and this is an Ames question. It's a very easy question, you know. If you just read the question, you don't even need to see what the cross is saying. The experiment shown in the figure has been carried out by Morgan, T.H. Morgan, father of experimental genetics, who discovered linkage, crossing over, etc., etc., to show the phenomena of linkage and recombination. If in cross 1, the genes are tightly linked and in cross 2, the genes are loosely linked, then what will be the percentage of recombinants produced in cross 1 and cross 2 respectively? So what is the answer to question number 12? Quickly tell me the answer. Read the question, okay? They are asking you about two different cross. One cross where genes are tightly linked, other where they are loosely linked. Then they are giving you 4 options, A, B, C and D. Good, I start getting the answers, but I want more. Take your time, it's okay. But once you solve this question today, you know, you will realize it's a very easy question. So I want answers from everybody. And if you don't know, just let me know that I don't know so that I can explain you, okay? Good, very good. I start getting more answers now. Perfect. So all of you are giving me the right answer, which is option number B. Because when the genes are linked, the recombination is low. That is why it is 1.3% for cross 1. And when the genes are far, the recombination is more, which is 37.2%. Very good. Next question. Which one from those given below is the period for Mendel's hybridization experiments? And they have given you 4 options. And you should know the right answer. And this question is not made by me. It has been asked in the NEET exam in 2017. Okay, you look at this question and you're like, wow, so many years. But if you know the answer, you know the answer. And again, I'm stressing, you know, I always tell my students, name of the scientists and years are very important. And that is the proof, you know. So let me know. Ah, good, perfect. I already got one answer, but I'm waiting for more. Very good. You guys are doing amazing. I'm very happy. Very good. You are very well prepared. And small things here and there, that is why, you know, we are doing such sessions. So that you can just quickly grasp what is missing. For example, that pedigree analysis question, you know, that is something that will give negative marking to a lot of people. but now you know how to approach those kinds of questions. Okay, I'm not getting more answers for this question probably. So the right answer is option number D. And for all those who don't know, please write it down. Write it down on a sheet of paper next to you. They can definitely ask you such questions. And it's a very easy question that can give you 4 marks, which will decide your rank. Okay, so just write it down. Good. So then let's proceed to question number 14. Again, assertion and reasoning question. Don't get confused with the options. Read the assertion, read the reason, write true or false, and then you will get the answer. So assertion in mirabilis, selfing of F1 pink flower plants produces same phenotypic and genotypic ratio. The reason is flower color gene show incomplete dominance. So what is the right answer? Question number 14. What is the right answer? See if the assertion is true, see if the reason is true, and then see if it is the right explanation or not. So what is the right answer for question number 14? I start getting some answers, which is good. But please, everybody of you try to answer. If you try to answer, even if it is a wrong answer, it's okay. I will not eat you through online session, and I'm a pure vegetarian. Just try to answer as much as you can. Very good, very good. Now I start getting so many answers, and I'm very happy when I get a lot of answers, and even happier when they are all correct. Okay. So selfing of F1 plant produces the same phenotypic and genotypic ratio of 1:2:1 because it is an example of incomplete dominance. Good. Next question. If a homozygous red flowered plant is crossed with homozygous white flowered plant, the offsprings would be all red flowered, half white flowered, all white flowered. So what is the right answer? Question number 15. Homozygous red flowered plant is crossed with homozygous white flowered plant. The offspring would be all red flowered, half white flowered, all white flowered. Quickly tell me the answer for question number 15. Perfect. Now I start getting answers. Very good, guys. You're really doing good. Good. Perfect. So all of you gave the right answer, which is all red flowered. And why this question was chosen, don't get confused that if they are talking about red and white, they are talking about incomplete dominance. If they are talking about incomplete dominance, they will write it down like they wrote it down in the previous question, either in Millabilis jalapa or in Snapdragon, or they will write down a homozygous red flowered plant undergoing incomplete dominance. Very good, everybody gave the right answer. Next question. Given below is a representation of a kind of chromosomal mutation. What is the kind of mutation? Is it deletion? Is it duplication? Is it inversion? Or is it reciprocal translocation? I hope you know the difference between all of these, and then you can easily answer the question. So what is the right answer for question number 16? Is it deletion? Is it duplication? Is it inversion? Or is it reciprocal translocation? Question number 16. I start getting mixtures of answers, which I also like, by the way. But yeah. Okay. Good. So, some of you are answering option C and some of you are answering option D. So one thing that you have to remember, guys, that reciprocal translocation is always between two different and non-homologous chromosomes. Okay, it cannot be the same chromosome undergoing reciprocal translocation. You always need two different chromosomes. Okay, there will be one part deleted from here that will go here, one part deleted from here that will come here. That is what is reciprocal translocation. So the right answer here is option number C, which is an inversion, because it is the same chromosome. You see, A, B, C, D, E, F, G, H. Now what is happening? It is A, D, C, B, E, F, G, H. And don't get confused why they have written it down here or why they have written it. Is just a way of representation of inversion. In a normal scenario, for example, if we represent the chromosome like this, we can say it is 1, 2, 3, 4, 5, and then there is inversion of 4 and 2 here. Then what will be the sequence? It will be 1, 4, 3, 2, 5. Okay, that is how they will represent, inversion on a chromosome. So don't get confused. Please remember this, that reciprocal translocation is always between two different non-homologous chromosomes. Okay, so the right answer here is option C, which is inversion. Some of you did answer, option number D. I hope it is clear for all those. Reciprocal translocation will always be between two different chromosomes, not the same chromosome. Good. Then let's proceed to question number 17. Again, assertion and reasoning, and this question has been asked 2 times, once in 2015, once in 2017. So maybe this is the year they will ask it again. Assertion is, in humans, the gamete contributed by male determines whether the child produced will be male or female. And then the reason they are giving is sex in human is a polygenic trait depending upon cumulative effect of some genes on X chromosome and some genes on Y chromosome. So what is the right answer? Read again, assertion carefully, see if it is true or false. Read the reason carefully, see if it is true or false. And then we proceed with what is the right answer for question number 17. Are they both correct, and is the reason correct explanation for assertion? If both are correct and reason is not the correct explanation, if assertion is correct but reason is incorrect, both assertion and reason are incorrect. So what is the right answer for question number 17? Quickly tell me the answer. Good, I start getting some answers. Very good. It's a very easy question. Yes, all of you are writing the right answer, which is option number C. Okay, in humans, the sex determination is by the Y chromosome and it is not a polygenic trait. Okay, it depends only on the Y chromosome. There is this testis determining factor that determines the formation of testis. Very good. Next question, number 18. Again, assertion and reasoning question. Assertion is aneuploidy may be of hypoploidy or hyperploidy type. The reason is monosomy lacks one pair of chromosomes. What is the right answer? You have to remember your lecture of genetic disorders, which is very important, and then you may be of hypoploidy or hyperploidy, and if monosomy lacks one pair of chromosomes. So what is the right answer for question number 18? Quickly tell me the right answer. See, as much as you practice, these assertion and reasoning questions, it will be very nice because they will ask you such kinds of question, in the exam. So just get used to familiarize yourself with such kinds of questions. Good, I start getting the answers. And the answers that I am getting is, should I tell before I receive all the answers? Good. So, I start getting the answer if both assertion and reason are correct, but reason is not the correct explanation. Okay. So let's go again step by step. Good. I see this, not sure though, which is good. I like that. So aneuploidy may be of hypoploidy or hyperploidy. This is definitely true, because what is aneuploidy? We have seen it is monosomy. Let's talk about the sex chromosome, Turner syndrome, and trisomy, which is Klinefelter syndrome. So hypoploidy is what? Turner syndrome. Hyperploidy is Klinefelter's syndrome. So this is true. Monosomy lacks one pair of chromosomes, and I am very happy because I see one right answer between the wrong answers. So monosomy lacks one pair of chromosomes is false. Why is it false? Monosomy does not lack one pair of chromosome. Monosomy lacks only one chromosome. Okay, because 2n minus 1 is what is. I hope it is clear for everybody. It is not one pair of chromosome, it is one. Next question. Good. So the next question, question number 19. Sickle cell anemia is? It is caused by substitution of valine by glutamic acid in the beta globulin chain of hemoglobin. It is caused by a change in a single base pair of DNA. It is characterized by elongated sickle-like RBCs within a nucleus, or it is an autosomal linked dominant trait. Good, I start getting answers already for question number 19. Sickle cell anemia is caused by what? Good, and all of you are giving the right, all of you are giving the answer option number A, which is the wrong answer. It is not the right answer. Sickle cell anemia is caused by valine by glutamic acid in beta globulin chain of hemoglobin. It is the wrong answer. Why? Because they have given it in other way round. In the normal condition, we have glutamate, and in the mutated condition, when there is sickle cell anemia, we have valine. So, it is caused by substitution of glutamic acid by valine, not the other way round. Clear? If it is clear, now try to answer this question again. So, for normal hemoglobin, which is HbA, we have glutamic acid, or also known as glutamate. And in case of HbS, which is sickle cell, it is valine. You see, that is how they will trick you. That is why it is important to do these rapid revisions, okay? These are questions, that we have chosen very nicely, so that you know that what is going on, okay? That is how they are going to trick you and easily here. So always be very vigilant. It is not caused by substitution of valine by glutamic acid. It is caused by substitution of glutamic acid by valine in the beta globulin chain of hemoglobin. Good. Now I start getting the right answer. It is caused by a change in single base pair of DNA because it is a point mutation. Okay, there is change in the DNA that ultimately changes the protein, the amino acid. Good. Then let's proceed to the next question. Again, assertion and reasoning question. Phenylketonuria is a recessive hereditary condition caused by body's failure to oxidize an amino acid phenylalanine to tyrosine because of a defective enzyme. This is the assertion. It is a long assertion, but read it properly, okay? They are asking you about this disorder, which is phenylketonuria. And it results in the presence of phenylalanine acid in the urine. What is the right answer? Question number 20. They are asking you about phenylketonuria, which is a recessive hereditary condition caused by body's failure to oxidize an amino acid phenylalanine to tyrosine because of defective enzyme. It results in presence of phenylalanine acid in urine. What is the right answer? Again, assertion and reasoning. Try to see if the assertion is true. Try to see if the reason is true. Try to see if reason is the correct explanation of assertion. And if you don't know, just quickly let me know. We don't know, so that we can proceed to the explanation. But it is a very straightforward question. You must have studied about phenylketonuria. Good. So a lot of you are answering that. Answer is, no, there are mixed answers. But I want some more answers. Okay, so some of you are answering option B if both assertion and reason are correct, but reason is not correct explanation of assertion. And some of you have also given the answer option number D, both assertion and reason are incorrect. So the assertion is phenylketonuria is a recessive hereditary disease caused by body's failure to oxidize an amino acid phenylalanine to tyrosine because of defective enzyme. This is true. The enzyme is phenylalanine hydroxylase. It is a recessive hereditary disease, okay, caused by body's failure to oxidize phenylalanine to tyrosine. And then it results in the presence of phenylalanine in uric acid, and this is also true. So both assertion and reason is true, but this is not the explanation for this. Okay, it results in the presence of phenylalanine in uric acid is kind of a result of phenylketonuria. This is not the correct explanation. That is why the right answer here is option number B. So all those who have answered option number D, which is almost half of you, is it clear? Okay, both are correct. You just need to go through, revise through these genetic disorders and see what are they caused because of. It is a very straightforward question. Okay.